Hello, I'm Dr. Claire Carvell. I'm a pollinator ecologist at the UK Centre for Ecology and Hydrology, and I coordinate the UK Pollinator Monitoring Scheme as part of the Pollinator Monitoring and Research Partnership. It's a real pleasure to be able to have a chance to talk for you today. Thanks to Hollis and Sam for the invitation, and I'm looking forward to hopefully joining you at the workshop. I'm going to spend the next 20 minutes just really trying to kind of tell the story of the UK Pollinator Monitoring Scheme, or POMS as we call it, right from our original remit and a design and testing phase through to the implementation of POMS as we have it now. Our story begins with the launch of national pollinator strategies across the countries of the UK and Ireland, so across England, Scotland, Wales, and a strategy for Ireland as well. And this was in recognition of declines in diversity and distribution and range of many of our key pollinator species, and also in recognition that whilst there was a lot of biodiversity monitoring activity going on across the UK, that was lacking in many areas for these critical pollinating insects. So as part of the National Pollinator Strategy for England, there were very clearly identified actions that involved gathering evidence. And these included to aim to develop and test a sustainable monitoring framework that could be implemented by professionals and volunteers. And that work began around 2014. And then following that, there were actions within the strategies to implement new monitoring schemes, so to establish recent and ongoing trends and their status with greater confidence than we had before. And there were further aspirations identified in these strategies involving improving data standards, expanding pools of taxonomic expertise, motivations of the volunteer recorders who would be critical to these monitoring schemes being long-term, and also supporting long-term storage of materials and new technologies. The two elements that we've been able to lead under the partnership that I'm talking from today are these first two elements at the top. And so the remit we originally had was to be able to design a monitoring scheme that would answer originally these two questions. How is the status of insect pollinator populations and communities changing over time in agricultural landscapes and in the wider environment of the UK? And secondly, to ask whether we could use such a monitoring scheme to answer the question of how pollination services to crops are changing over time. The spatial scale was to be operating across the UK, but to have an ability to look at detecting changes at the country level, so across the different countries, and also to focus on wild bees and wild hoverflies, of which we have around 260 to 280 species recognised of those two taxa, but to choose approaches that would also sample, of course, the massive range of other flower visiting insects, including the honeybees. The approach we took for this framework, if you like, was to have a common set of themes running through our thinking. We wanted to start by being really focused on the outputs or metrics that we'd be intending to generate as part of this monitoring. And those outputs might include various different measures of pollinator population status or health, such as abundance, distribution, diversity, or measures that relate to the pollination service provided, or indeed the capacity of the landscape or the quality of the landscape to support pollinators. And in order to have a monitoring scheme, again, which is sort of well structured and well designed, we wanted to think in terms of the components of this framework. So the recorders, those that would be required to collect the information, whether they, and we need to understand what the sorts of expertise, whether they're volunteer or professional, in order to be able to understand the costs of such a scheme. We thought carefully about the methods that might be employed, either applied on their own or in combination to deliver more robust metrics, about the sampling site networks, so networks of sites to sample that might range from ad hoc opportunistic random sampling to much more systematic or systematically located sites. And of course, to consider the support tools that be required to underpin and support such a scheme, including data validation, analysis, storage of materials, ID guides, and so on. So with that in mind, we set up a series of workshops and consultations with both the sort of wider NGO community, charitable trusts, and particularly also entomologists and professionals involved in collecting data on bees and hoverflies and other insects across the UK. And this was in order to collectively try and assess the capacity of these different methods and combinations with different types of recorder to provide us with data on these different metrics of interest, or indeed to consider how we would tackle this challenge of taxonomic resolution of the data collected. So we had these kind of framework tables where we were effectively scoring the ability of a particular method, in this case pan traps, in combination with whoever was either collecting or processing that information to generate metrics of abundance or species level diversity for the different taxonomic groups of pollinators. And we then took some of the, I guess, best methods and ideas and concepts from that stakeholder exercise out into the field. So we conducted a replicated test of different methods across what we call the wider environment at various different sites in England, north and south of England, comparing pan traps with transect walks and timed focal floral observations as the three key methods. Within that, we also did some smaller experiments looking at differences in duration and bowl size, for example, or width of the transect that was undertaken. And we wanted to apply this in a way that would be practical to adopt as part of a long-term monitoring scheme. So we operated this, what we call a one-day, one-person protocol, to allow repeated site visits to a particular area. In this case, a 1 kilometre square site is the sort of spatial scale of the site. And in particular, we wanted to test the ability of these methods to generate the sort of sample sizes that would be required to assess trends in abundance of the common pollinator groups and potentially species over time. And I'll come back to that element of sample size in a moment. So by comparing in this pilot study different sampling methods across different pollinator groups and species, and also looking at the data that was collected by both volunteer recorders and expert taxonomists in the field, we came up with some quite interesting insights, which allowed us to then really design the scheme that we're now implementing. So, for example, these species accumulation curves taken from both pan traps and transects at the same sites, which are reported in the O'Connor et al. Paper in Methods in Ecology, you can see that they're relatively level pegging in terms of the sort of general abundance and species accumulation, but typically pan traps are more consistently sampling a greater number of species, so the species richness is higher. And of course, recognising the real difficulty with observational transects in terms of identifying insects on the wing, we've also then got some interesting insights across those different taxonomic groups with the impact of having experts out in the field rather than potentially researchers or technicians. So again, the ability of an expert undertaking a transect is probably likely to sample an equivalent species richness or species community as a pan trap. And of course, then we have to think of the practicalities of having those experts across a large number of sites in the field. So this is an example of the kind of framework that we came up with in terms of, again, sitting around and collaboratively looking at all these different possible combinations of recorders, methods, site networks, and on the right hand side of this slide, the different outputs that would be generated from different designs of scheme. And these are sort of kind of discussed and presented in the Journal of Applied Ecology paper by Tom Breeze and the rest of the partnership. At the same time, in this design and testing phase, we were able to use both a sort of consultation of pollinator experts, but also some existing research project data to ask questions about power and sample size. So we wanted to be able to design a scheme that had at least 80% power, statistical power, to detect changes in defined response measures over a period of 10 years. And so this is just an example here of where, if we're looking, for example, to have a 30% detect change in a metric of 30% change or an annual change of 3.5% over time, with a typical kind of initial annual counts in year one of that scheme of one individual or an abundance of one, we would need up to 145 sites in our total network in order to understand trends in these kind of metrics of these kind of pollinator groups. If we have greater sample sizes at the start, so if our mean count in year 1 was higher, up to 10 individuals, then we would need fewer sites. And as you'll see in a moment, it's this level of detection, detecting annual changes of 3.5% with average counts of 10 individuals across 75 sites, which is the sort of level that we've actually gone for with the UK POMS. So this takes us, to the birth of the Pollinator Monitoring Scheme. Again, it's been a real pleasure to be part of this kind of journey. The scheme was actually implemented in 2017 and really has been the first in the world to actually be able to generate systematic data on the abundance of bees and hoverflies and this whole range of flower visiting insects at a national scale. What we're doing is critically, we're not ignoring the wealth of long-term species occurrence records that we have for the UK, and we're very much integrating those into the current scheme. So it's combining these long-term records of species occurrence at individual sites with these 2 large-scale surveys to try and track changes in populations. And the 2 surveys I'm going to try and briefly talk to you and tell you about that we are now running are effectively timed observations, so flower insect timed counts, or FIT counts as we call them, and we have a much more intensive systematic 1 kilometre square survey that runs across the UK. And still, in line with our original remit, this scheme is underpinning the evidence base for the national pollinator strategies of England, Wales, Scotland, and now we have Northern Ireland joining in with POMS for the first time in 2021. So the flower insect timed counts were designed really as kind of, if you like, our real big citizen science activity, and they're aiming to collect data on the abundance of flower visiting insects, particularly understand more about plant-pollinator interactions, and to do this across a variety of habitats and places. So they can effectively be done anywhere, in gardens, in parks, in schools, in the countryside, on crops, in a whole range of habitats. And we have a few simple weather criteria, so they need to be conducted in warm, dry weather, and we have some temperature thresholds based on ideal conditions for insect activity in the UK. And the way the protocol works is that you count all the insects that land on a patch of target flowers within a 50 centimetre square patch during a 10-minute period. And importantly, the insects that are counted in that 10-minute period are identified to group level, so separating bumblebees from honeybees, from wasps, from solitary bees, butterflies, and so on, rather than trying to identify to species. And our data entry system allows people to submit photographs of those groups, so we have a means of validating what they're counting. The target flower list is quite carefully chosen to kind of try and maximise the sample sizes across this set of flowers. And these are effectively common and widespread plants that occur both in gardens and across the UK countryside. So as you can imagine, this kind of citizen science activity was quite heavily impacted in 2020. We had a lot of group activity going on previous to that. For example, in 2019, lots of group activities in the London parks and in some of our nature reserves. And we even have collaborations in Chile with colleagues who are exploring the FIT counts as a way of conducting similar kind of monitoring methods in Chile. So that was 2019 was a great year. We then, of course, had a lot of us confined to just doing the counts in gardens, and later on, when we were allowed out of our lockdown, into these more widespread countryside areas. But the effectively the point is that this actually provided a really fantastic activity for people to connect with nature, really on their doorstep during what was a really tough time. So you can see here the impact of, I guess, the promotion that we've been doing over time, and the counts gathering momentum. So going through from the first year where we had a pilot, 2018 was the first year in which the FIT counts were rolled out nationally. Northern Ireland came in only in 2020, so the last year on the plot here. But we've ended up with a real boost in some of the early counts. This is indicative of what happened in our lockdown in England last year. So in this set, we do have probably about 70% of the counts coming in from gardens, but a good number coming in from the wider countryside, and a good number of people taking part, and that's been increasing year on year. What we're trying to do with these data is partly just be able to visualise the different kinds of insect communities that are visiting the different target flowers across different habitats, and how those communities, groups of insects and numbers might be changing over time. So this is one of the graphics we've developed, which seems to be a really nice way to explain how the fit counts are panning out. And as an individual user, you can see how your own fit count looks, and you can then be presented using the visualizations that we're developing with how your picture as a recorder might compare with the national picture, for example. So ivy is a common plant that occurs in gardens and the countryside, and here's an example of a plant that attracts actually quite high numbers of wasps, so quite a high proportion of wasps and flies in the communities that are visiting ivy. Some other examples: hawthorn is an early flowering hedgerow plant in the UK. You can see again the flies appear to be really quite important visitors here. We've also got the solitary bees coming in, so it's an important source of early nectar for them. And the lavender, of course, is a really popular garden plant. That can be grown in pots, in containers and in borders. And here you can see again that really clear signal that it's the bumblebees that are tending to visit the lavender in larger numbers, and also the honeybees. So these are just some of the ways in which we're analysing and looking at the data. And of course, we're also running models to look at effects of landscape type and changes over time as well. We are managing to collect some feedback from the participants of the fit counts, which is really important, I think, in developing citizen science schemes like this. So just as an example, people are generally finding it relatively fun to do a count, and also not too difficult. So the exciting thing for us this year is that we've had some additional funding in order to develop a new app, and this is now available to download. So the Fit Count app is on both the Android and the Apple stores, and it provides, I think, a really nice kind of accessible way to do the count, with also some embedded guides for differentiating the different insect groups. And we're also having to translate the app into different languages. So here's an example of one of our Welsh pages to support our Welsh contingent, and recognise the funding we received from the Welsh Government. And we're also really excited to be developing the app in other countries, parts of Europe and also South America. So I'm just going to move on in the last couple of minutes to tell you about our intensive systematic survey. This is set up over a series of 75 1-kilometre squares across Great Britain, and will be extending to Northern Ireland this year, and it covers both semi-natural and agricultural habitats. So the squares are stratified such that they represent the relative covers of those different broad land use types across the UK, and again is designed to detect UK or GB level changes in different pollinator groups and in some common species. But again, we recognise that the abundances that we're catching may not be suitable enough or substantial enough to track trends in all species from all the sites. We have co-located our sampling with other long-term national environmental monitoring schemes, again, which allows some really useful co-located data to try and help explain some of our results. So the survey works by having a series of pan traps set out across the 1-kilometre square that you can see in each case here, and is a typical map that our volunteers or our surveyors would have. And the way we've set this up is that the survey is conducted 4 times each season to try and get the sort of data that we want to generate. And the samples are sent back into us in the labs at CEH Wallingford. I'm going to go quickly through. You get an impression that with a large network of sites, we've got some really fantastic locations. This is our survey team, and these guys are working as mentors to a team of about 55 volunteers who are set up across this different network of squares, and who receive all the kit and all the sample processing back up from us back at base. I'm going to go quickly through my last couple of slides now because I'm realizing that I'm getting out of time. So again, just to show you some of the highlights that we like to provide to our volunteers and recorders, some of the numbers there that we report, and again have to do in Welsh as well. We regularly share updates on the squares that are available to adopt, and again lots of equipment and training provided. We're also really excited to be undertaking some quite sophisticated integrated modelling in order to try and combine the data that we get from these structured surveys with the unstructured surveys of species occupancy, and that gives us a much better idea of usually the precision of our estimate, particularly of distribution of species over time. And again, it's great to work with some of our partners on that. So I'm just going to finish by very quickly glossing over some of the slides on the impact that we've had, the impact that we're very proud to be having in some of the other countries. So as I say, we're working with Cyprus and South America. And I've got a slide here which really, I hope, just encapsulates a summary of some of the opportunities and also the challenges that I think will always be faced by these kind of monitoring schemes, particularly where you have tricky taxonomic groups like the bees and the flies and these small insects for which we have such a small core group of expert taxonomists. So I'm going to wrap up my talk just by acknowledging our funders and partners and volunteers on the Pollinator Monitoring Scheme, and thank you very much for listening.